Hello and welcome back to HeFPipe. At this point, we're going to talk about testing for single locus associations, or SLAs, with an F-ratio test. So in the pipeline, after all the correlation analyses have been run, which they have been in the last video, you're prompted to test the dataset for single locus effects by comparing a model that invokes heterozygosity at each locus as an independent predictor of a particular trait to a model invoking only the multi-locus heterozygosity estimator as the singular response variable. So you have one model that has lots of different response variables representing each locus, and another model with one response variable that's a composite of all of the other loci. Now, if you want to perform this analysis, you'd be prompted here, and you just say yes by typing Y or typing yes, and then hitting enter. Then you're prompted to list each trait for which you would like to perform the test. So if you want to look for single locus associations with several different traits, you would list those traits. And you can see here that the trait options are listed right in the terminal. You type as many of them as you desire, pressing enter between each of them, and then when you're done, you type done. So let's say I want to look for single locus associations for queen weight initial, which is one of our fitness traits in the example data set, and survived eclosion. Okay, those are the only two I care about, so I type done. And then the pipeline prompts you to enter the multi-locus heterozygosity estimator to be used for the model comparison test. So we recommend using MLH, but you also have the option of using IR, or internal relatedness, SH, which is a standardized heterozygosity, or HL, which is homozygosity by loci. I'm going to use MLH. And again, if you're not sure what they get called in the data set, they're listed right here, 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 and here. And then I type enter. At this point, you're prompted to designate an error distribution and link function for each trait being used as the response variable in the model comparisons. So the error distribution and link function designation is used in generalized linear models to relate the response variable, which, because they're generalized linear models, is allowed to be non-normally distributed and to have a non-constant variance to the predictors. So the link function basically allows the response variable to relate to the predictors. If the response variable is normally distributed with constant variance, the error distribution is the default Gaussian distribution with the default identity link function, and you should just type Gaussian. Okay, this happens to be the case for our first example here, which is initial queen weight. I know the distribution of initial queen weight is pretty much normal, so I type Gaussian here. Now, if the response variable was, say, binary, which we'll get to in a second, the error distribution would be binomial, and the default link function would be a logit link function, and you would just type binomial. Note at this point that the pipeline is actually using R to do some of these linear model calculations. So this GLM function performs the regression analyses at this step of the pipeline, and it can accommodate other families besides Gaussian and binomial, for example, Poisson, and I'll provide a link in the video to R's documentation about the different error distributions you could use. So the pipeline prompts you to designate an error distribution separately for each trait that you entered, and we'll see this in a second, and it lists the traits before it prompts you to enter them, so you'll get reminded which trait you're trying to designate an error distribution for. So you'll notice I typed in Gaussian for initial queen weight, and this time for survived eclosion, I'll type binomial. And I'll hit enter. So the test or tests you just conducted generate 4 types of output files. The first is called fitness and loci combined.csv, and that is located in the single locus effects test subdirectory. And here we are, fitness and loci combined.csv. And this file is basically a combination of all the data from MLH output.csv, which we've seen in an earlier video, which is basically just all of the fitness data for all of the samples that get analyzed in this particular run of the pipeline, so all the samples that aren't on rejected list, as well as the multi-locus heterozygosity estimators here. And then it combines all of that with all of the data from heterozygotes and homozygotes.csv, which again was generated earlier, and again basically has all of the genotype information for all of these samples. So this file is a combination of all of the fitness traits and all the loci. Except in this case, instead of being exactly a copy of heterozygotes and homozygotes.csv, all of the NAs in heterozygotes and homozygotes.csv have been replaced with the mean heterozygosity at that locus for the samples that were typed. So whereas this is missing data, for example, these two or all of these are missing data in heterozygotes and homozygotes.csv, here they get replaced by the mean across all samples that do get scored at that locus. And the reason it does this is because this is what was recommended for the single locus association test in the original documentation, the original articles. The second type of output is generated for each trait that you entered in the beginning of this module and is a text file that's called a multiple regression versus single regression, and then your trait name here. So in our case, we have a multiple regression versus single regression. Let me see if I can. For queen weight initial and for survived occlusion. So let's open the queen weight initial file. And it contains all, you can see that it contains all of the information regarding the significant predictors for each of the two models described. So here's model 1, and here is model 2, as well as the F-ratio test comparing the two models. So the significance of this F-ratio test is displayed as the last line of each text file. So here is the p-value of that significance test. And if the p-value suggests that the F-ratio test is significant, the meaning of this is that the two models are different, and it provides support for the existence of a single locus association for the trait in question. So what we can conclude from this output, because the p-value is greater than 0.05, is that we failed to detect a significant difference between the two models, and therefore we failed to find support for any particular locus bearing a single locus association at the trait queen weight initial for our panel of loci. The third type of output is again generated for each trait that you entered in the beginning of the module, and it is a TIFF file that's called multiple regression trait name here. So we'll take a look at this. This is the TIFF file, and it contains a plot, or it contains a series of plots that help you decide whether the assumptions of linear regression are satisfied for the model that you parameterized by each locus treated as a separate predictor. Note that interpreting residual plots for models invoking non-Gaussian error distributions is not always appropriate or straightforward. So here we have a model that does invoke a Gaussian error distribution. So interpreting these plots would be fairly straightforward and appropriate. And you can see here that there doesn't seem to be a lot of deviance from assumptions of linear regression. So this is probably a kosher test to have performed. Taking a look at survived eclosion, which remember had a binomial distribution, not a Gaussian, you see that there's all kinds of funky stuff going on with these plots. But you can't really interpret these plots straightforwardly, so it's not clear based on these plots whether the assumptions are violated or not. The fourth type of output is again generated for each trait that you entered in the beginning of the module, and it is a TIFF file called single regression and then the trait name. So we have, because we entered two different traits in our analyses here, we have two different files of those. One's for queen weight initial and one's for survived eclosion. Let's look at the queen weight one again. And again, this contains plots that help you decide whether the assumptions of linear regression are satisfied for the model parameterized, but this time only for the single locus heterozygosity predictor. So this is MLH. This is not every single locus as a separate predictor. Note again that interpreting residual plots for models invoking non-Gaussian link functions is not always appropriate or straightforward. So that's it for the single locus association F-ratio test module of the pipeline, and these are all of the output files that get generated. And again, these are a good complement for the GFAST analyses that we did earlier. So both these tests and the GFAST analyses look for single locus associations with particular trait values. In this case, it tells you whether to expect them at a certain trait. In the case of GFAST, it actually singles out which particular locus might be responsible. That's it. Stay tuned for a continuation of the pipeline.